being here. I'm excited to talk to you about next generation immuno-oncology biomarkers. This will be a little bit of a different perspective than probably what you've heard. I come from a consulting background, and we focus primarily in the diagnostics and research tools industries. And so we spent a lot of time and done a lot of research and analysis over the past 2 years, helping our clients understand where are there opportunities for diagnostics in immunotherapy. And so what I'll share with you today is some of the analysis that we've done and some of the takeaways and the conclusions that we've drawn from some of our research on next generation IO biomarkers. I apologize in advance, a lot of my slides are going to be somewhat text heavy, so if there's something I skip over or don't go into in too much detail, I'm happy to send the materials to you afterwards. And disclaimer, some of the companies listed may be some of our clients or customers. So I will start with the takeaways, and based on some of our research, which has included an analysis of about 1,000 IO clinical trials, as well as discussions with experts both from the clinical and the industry side, we've identified just a handful of takeaways that we'll discuss today that we think are important for diagnostic companies to consider when thinking about the future for IO diagnostics. The first, which is probably not surprising to anyone, is that the IO biomarker landscape is rapidly expanding beyond PD-L1 and MSI. I think most people here already know that, but we'll provide some context to that and quantify a little bit. The second message is that we think IO is going to drive significant changes in terms of the types of technologies that are being used for cancer diagnostics, as well as how and where these diagnostics are being performed. And the third is we think there's an opportunity for immune response monitoring that is to some extent being overlooked. And so we'll discuss what we think is a fairly attractive opportunity for diagnostic companies in immune response monitoring. Just to give you some background about the analysis that has gone into some of the points we'll talk about in this presentation, one source that we use is a biomarker database that we've been building and maintaining for about 1.5 years now. It has data from over 1,000 IO clinical trials. The way we build this database is we go through clinicaltrials.gov, we identify immuno-oncology trials, and then we filter that down to focus just on the trials that are discussing biomarkers or are using analytical technologies to measure some kind of biomarker. To date, we've identified about 1,400 active and ongoing trials. We've analyzed about 1,000 of those. We then extract as much biomarker information as we can. So we're pulling information about the trial, the phase, start date, enrollment, sponsors, as well as the biomarkers themselves, what technologies are being used, the sample types, basically anything we can find. We also cross-reference the NCTs with conference abstracts because there's a lot of rich data being presented at conferences that isn't necessarily found in clinicaltrials.gov. So we really try to get as much of a comprehensive picture around these biomarkers as we can. We then aggregate everything into a data visualization tool called Tableau, which allows us to segment the data and filter it and identify trends. For the sake of this talk, we also wanted to interview key opinion leaders, experts in the IO space to get their perspectives on next generation IO biomarkers, to understand how are they using them, what do they see as trends, how do they think the future looks for the IO space. And so we talked to 15 stakeholders, both from the clinical and academic side, as well as the industry perspective, and we have US and European perspectives here. You can see some of the affiliations of the stakeholders that we talked to. I'll note that this is a small number. We usually do much bigger interview campaigns, but we think it's enough to give us some insights, at least directionally, in terms of interesting trends and identify interesting areas for further research. So that leads us to the first point, which is that the IO space is rapidly expanding beyond PD-L1. This chart here shows all of the biomarkers that we identified in the 1,000 or so clinical trials. Each bubble corresponds to a different marker, and the size of the bubble corresponds to the number of times that or the number of trials in which each marker is mentioned. So you can see there's a lot of activity. We identified about 4,600 mentions of at least 1,200 different markers. And when I say a biomarker mention, what I'm referring to is basically a distinct reference to a combination of a biomarker, a testing purpose, a technology, and a sample type. So many trials will mention even the same marker in multiple contexts, and so we distinguish the different ways that each biomarker is mentioned. The color in this figure shows the different types of markers. So the landscape is primarily protein-based markers. About 40% of all the markers we identified were protein. Nucleic acids represented about 25% of the markers. Cell-based markers were another 12. The gray corresponds to combinations, typically cell protein combinations, which are about another 12%. And then everything else was either not specified or something else. And what we think is interesting is we track these trends over time, and we've noticed that since 2014, the number of biomarker mentions in each trial has grown. In 2014, it was about 4.5 mentions per trial, and it's grown to almost 8. So we see that more and more people are exploring not only just new biomarkers, but even the same biomarkers in new ways, using different technologies or for different purposes or in different sample types. This shows some trends for the top 15 biomarker classes or biomarkers over time. These top 15 biomarker classes comprise about 65% of all the biomarker mentions. You can see the top 15 that we called out here. PD-L1 is still the largest marker, which is not a surprise, but you can see that there are a handful of other markers that also have substantial representation. For predictive purposes, the top markers that we've identified other than PD-L1 are tumor-infiltrating immune cells, peripheral immune cell populations, gene expression profiles, PD-1, and tumor mutation burden. From a monitoring perspective, we see that peripheral immune cells, cytokine profiles, and gene expression profiles are being widely used. And then in terms of the fastest growing markers based on the number of mentions, PD-1, TILs, CTCs, tumor mutation burden, and gene expression profiling have all been growing at about 80% compared to 70% annually for the rest of, or the overall biomarker landscape. One of the things that's always surprised us about the immuno-oncology landscape is that it develops much more rapidly than we expect or anyone we talk to expects, and we expect that to continue. In terms of next generation biomarkers, we see a lot of opportunities for development, even just in the next year. This shows a selection of next-gen biomarker trials that have primary completion dates in 2018. So this is not a comprehensive list. We actually just filtered this to focus on trials that have at least 100 patients enrolled. And you can see that there are, I think this is at least 24 trials that are expected to yield biomarker or next-gen biomarker data in 2018. And we called out a few ones of interest here. This Roche Empower trial is looking at gene expression profiling. The Pfizer ovarian Javelin trial is looking at CD8 positive TILs, FOXP3, PD-L2. The Merck Keynote 181 is looking at gene expression profiling as an exploratory marker, and this NCI trial is looking at tumor mutation burden. And note that these are all phase 3 trials with pretty decent enrollment, so we think there could be some good data coming out. When we talk to stakeholders about next-gen biomarkers, asking them what are their perspectives, most of them anticipate that tumor mutation burden and tumor-infiltrating immune cells are going to be the markers that are likely to have the most significant clinical impact in the near term. What's interesting, and I think this echoes some of what we've heard from Ken and Kim, is that a lot of these markers are just different ways of assessing the tumor microenvironment inflammation status. They fall along different paths of that inflammatory pathway that we've seen a few times. And so it will be interesting to see which one of these different technologies or markers rather becomes favored or sort of drives adoption for measuring the tumor inflammation status. Some of the feedback on TMB is that people like it. It's been in multiple trials. There's been some data around TMB, and so they expect that to be extremely relevant. It's pretty accessible. A lot of people are already getting TMB results on their foundation reports, and the report is objective. It's quantifiable, and they like that. For tumor-infiltrating immune cells, people see this as the most direct and the simplest way for measuring tumor inflammation. You can do this just via H&E staining or IHC. However, the particular cells, the markers, and the cutoffs for measuring inflammation still need to be determined. And this is also a more subjective readout. Gene expression profiling, we've received some mixed feedback. People, I think, are really excited about it. They're still waiting for more conclusive data. I think in standardization of the signatures, there are a handful of different T effector signatures that are being explored, and no one's really quite sure which one's most relevant. There are a lot of technologies that are being used to explore this, and so there are a lot of opportunities to bring this in-house, and I think people find that interesting. But I will note that I think just this morning there's been some interesting data from the Roche Empower 150 trial using a gene expression signature, so there are developments being made here. Some of the other markers that are discussed by stakeholders are the other checkpoint targets. I think a lot of people anticipate that some of these other assets are going to go in the same direction as PD-L1, and that those checkpoint targets will be included in any sort of diagnostics that are associated with the therapies. And then there are a handful of monitoring markers, T-cell receptor clonality, as well as cytokine profiles that people are exploring primarily in early stage trials that they think look promising as well. But I think what's one of the more interesting takeaways that we've heard when we talk to people is that there's no single marker that's expected to be used exclusively. Almost everybody anticipates that eventually all of these, or multiple different markers and even different diagnostic modalities are going to be combined to select therapies or IO therapies. Moving on to the next point, which is about technology. As I mentioned, we think that the IO landscape is going to change a lot of the cancer diagnostic technologies. And so here's a summary of the technology breakdown from our Immuno-Oncology Biomarker Database. You can see that immunohistochemistry remains the most widely utilized technology, represents about 40% of the biomarkers in our database. 19% is flow cytometry and about 12% sequencing. However, the sequencing numbers likely underrepresented because a lot of the biomarkers that are mentioned in trials don't actually mention a particular technology, and those tend to be genetic markers, and so we think sequencing is being used a little more than this. However, if you look at the growth, we do see that sequencing, which is this medium blue here, is experiencing the fastest growth in terms of utilization within these clinical trials. And so we do expect to see quite a bit more sequencing in the future. There are not many platforms that are mentioned by name in the trials, but we like to call them out when we can. The trials, or the platforms that are mentioned most commonly would be the NanoString for gene expression profiling, Luminex for cytokine analysis, Foundation Medicine for TMB, Fluidigm for gene expression, and CyTOF, as well as CellSearch for CTCs. And so this is the interviewee feedback or the stakeholder feedback on technologies. And I think what's interesting here, and I think some of this was touched on in some of the talks yesterday, is that stakeholders indicate only a relatively small share of labs are equipped to conduct next-gen biomarker analysis in-house. And they think that even in the future, not a lot of labs are going to be equipped to do this in-house. These two columns show the interviewees, the feedback from the interviewees that we spoke to, which primarily represent large academic medical centers. So they're not necessarily reflective of the entire oncology diagnostics market. But we asked them about what do they have the capability to do in-house currently today. And they indicate that they can do IHC, flow, cytokine profiles via ELISA, and for the most part NGS, primarily for tumor mutation burden. Other types of analyses, gene expression signatures, they either don't have access to the platforms or the informatics capability to do some of that. And then for some of the other markers, liquid biopsies, multiplex IHC, little capability to do that in-house today. There is some appetite to bring in some of these, particularly around TMB. We see that there's some appetite to bring that in-house, as well as multiplex IHC. There's a lot of interest in being able to do that internally. But other than that, we see moderate or low interest to bring in some of these technologies in the future. And then when we ask them about the community setting, because we know that most cancer patients are diagnosed and treated in the community setting, not in academic medical centers, they indicate that outside of IHC, the community setting is not equipped to do a lot of these, or if any of these at all, and that they are, even in the future, likely to be willing to keep that outsourced. And so we think that's important for a lot of diagnostic companies to keep in mind when they think about the next-gen IO biomarkers. Here's some of the insights around the centralization or the decentralization. I won't read through all these quotes, but about 2/3 of the interviewees indicated that they see IO biomarkers driving centralization of testing. And the drivers for that include lack of in-house expertise in terms of oftentimes bioinformatics, interpretation or scoring of these more complex assays, lack of test request volume. A lot of these are still relatively low volume assays, especially if you think about smaller institutions or community hospitals. And for a lot of these, it will save money in the near term to send these out. It takes a lot of time, energy, and resources to acquire an instrument, train a team, validate a test, and so in the near term, it's just easier and cheaper to send some of this out. There was some feedback that this is mostly actually from the larger, more sophisticated academic medical centers, that there is an interest and there is rationale to actually bring this in-house. And the main drivers for decentralization of testing or bringing this in-house are reduced turnaround time, which we hear a lot, and especially for these more complex assays that take 3 to 6 weeks to get a result, that's just too long in a lot of cases. And so there is some desire to bring these tests in-house to reduce that turnaround time. Many institutions have a preference for internal processes. They like to just have more control over the diagnostic process. They really like to keep control over the sample to do additional research. And then lastly, ultimately, for some of these institutions, if the reimbursement scenario is right, it could be less costly to do this internally or even profitable for them. And so there are drivers both for centralization and decentralization. It varies a lot on the institution. Lastly, I will discuss briefly what we see as an interesting opportunity in immune monitoring. And I see I have 5 minutes, so I'll go through this relatively quickly. But most of the biomarker work being done today is still predictive. About 40% of the biomarker activity is still being done for predictive purposes, but we do see about 25 or 26% being done for monitoring purposes, either immune monitoring or drug response monitoring. And there are a number of different ways that people are going about immune monitoring. Immune cell populations and phenotyping by flow cytometry or sequencing is probably the most common method. There's also antigen-specific immune responses by interferon gamma release assays or ELISpot assays, peripheral cytokine analysis, or TCR by sequencing or PCR. When we talk to stakeholders about monitoring, it's interesting that there's actually a lot of interest in translating monitoring from the clinical trial setting into clinical practice. And about 9 out of the 15 people that we talk to think there's a high likelihood that in the next 5 to 7 years, immune monitoring assays will become part of routine clinical care. The main drivers for that are for informing the timing and the frequency of doing scans, for distinguishing between pseudo-progression and true progression, or identifying hyperprogressors before that first scan, which oftentimes doesn't occur until 2 or 3 months, managing adverse effects, and adjusting treatment strategies. They mentioned when we talked to clinicians that they would adopt a monitoring test or utilize a monitoring test fairly frequently in the first 3 to 6 months of treatment, anywhere between any 2 to 4 weeks, followed by every 8 to 12 weeks after those first few months. And then one other interesting note that was brought up is that some of the KOLs anticipate that there might actually be payer willingness to reimburse these monitoring assays. If you think about some of the new reimbursement strategies that are focused on response or efficacy, there could be a rationale for payers to drive adoption of monitoring to provide an earlier readout of response. And so there's some thoughts that some of these models that are being used with Kymriah, for example, could extend to other immunotherapies. As consultants, we always like to think about market opportunities, so I would be remiss if I didn't at least show a high-level market analysis of what the opportunity could look like. This is sort of a really high-level back-of-the-envelope calculation, but if you consider sort of the incidence of metastatic cancer in the US and Europe, different rates of treatment with immunotherapy, different rates of adoption with monitoring assays, different frequencies, and the different distributions of different technologies, we think that the market could be anywhere in the range of 65 up to over 100 million. And this doesn't sound like a lot when you think about the dollar amount in terms of what the drug sales are, but from a diagnostic perspective, it could be an attractive opportunity. So I'd like to end actually with some key questions that we think this analysis leaves for diagnostic companies. The first is, how do established cancer diagnostic companies, those that focus primarily in IHC and PCR, adapt to a landscape that's likely to become increasingly complex from a technology perspective? The next is, how do DX kit and instrument manufacturers that rely on a distributed cancer diagnostics model adjust to a landscape that's becoming increasingly service-oriented? The third is, how do diagnostic companies position themselves to capitalize on immune monitoring? Next is, how do next-gen biomarker technology companies differentiate? There are a lot of companies out there. How do they differentiate to become partners with pharma companies? And then lastly, how do DX companies prepare for increasing integration of different markers into a single readout, so combining genomics, proteomics, and cellomics? And so with that, I'd like to thank my colleagues who contributed to this work, Tom and the Planet Connect team for having me here, and I'm happy to answer any questions.
Drew's slides.